So I don't know if anybody took a look at this, but I found this a while back and then actually had completely forgotten about it. But this is from a website called Visual Science. They have a bunch of this stuff for different viruses or different biological things, I suppose. Here is a video of, if we can get this to play, a video of the virion itself. So this is showing genome. L1 and L2 are the capsid proteins. L1 is the major capsid protein, and L2 is the minor capsid protein. We'll repeat that. And I wanted to show, I think, this one. Yeah. So this is looking at a cross-section of it. So this is showing genome in the middle. Note that the genome is bound to something. Anybody know what that might be? Exactly. It is histones. So because this is found, ultimately what we find is that this genome localizes itself, bless you, to the nucleus. It will bind itself to histones. And so in very much the same way, it's packaged around histones like eukaryotic chromosomes are. And we'll find those histones actually become bound then in this capsid as well. The only proteins that are in this capsid then when the virus exits from the cell are those histones and then the L1 and L2 proteins that make up the capsid structure itself. These self-assemble. The vaccine for HPV, so there's now the non-valent one. Those all are vaccines that don't include any genomic material at all. All it includes is L1 and L2 protein from those different strains of HPV. Okay. So the goal of those vaccines is to recognize the external structure, not necessarily what's happening inside of the host cell. And here's a different version of this. I think the one thing I want to point out here is the fact that these L1 proteins then end up forming the spikes that are characteristic for attachment. All right, that's about all we're going to talk about the structure. I bring up this slide here, the host cell hurdles, because I want to remind you of them. So some features that exist for host cell hurdles. Competition for the ribosome, actin remodeling. This virus does do some actin remodeling. We're not really going to spend much time talking about that, but it does happen during late stages of infection. The ones that we're going to actually spend more time with here are availability of proteins that support viral replication, and then compatibility of viral mRNA with the ribosome. Okay? And we'll focus on the second of those two more so today. The first one, replication, we'll talk about on Wednesday. What's a eukaryotic mRNA look like? What are the key characteristics again?
5' cap and poly-A tail.
5' cap, poly-A tail. Linear, yeah. How about so this would be the form of a mature mRNA. What about an immature, or what we sometimes call a pre-mRNA? What else might be in here? And notice I've drawn this a little bit longer. Yeah. We might have introns, we might have exons, etc. Here's the HPV genome. 8 proteins in all. E stands for early, so these are all ones that are expressed early on in in the life cycle. L's stand for late. This is not a perfect separation because even with those earlies, there are some that are more early than others. For example, one of the first expressed is this protein right here known as E2. Okay. We'll even find some of those early ones expressed late in late stages of infection as well. Probably the most important way to separate this, though, is that L1 and L2 are structural proteins. They make up the capsid. The E proteins, 1, 2, notice there's no 3, 4, 5, 6, and 7. Those all are non-structural proteins. So they're only expressed during the production of, during the production of the viral particles. Couple of things I want to point out here. There's a region known as URR. Our book refers to this as an LCR, which stands for Long Control Region. URR, Upstream Regulatory Region. This contains the origin of replication. So that's where replication obviously would begin from. It also contains the promoters to drive transcription. There are 3 promoters and they're all located right here. Transcription for this always occurs in the clockwise direction as is shown. There are 2 things there that are labeled A sub E and A sub L. Any guesses on what those are? Take a look back at our mRNA if you need to. Tail in particular. So A sub E would be the poly A signal for transcription from promoters for these early genes, and then A sub L is for a second transcript that would also produce these late genes. Okay. Anybody see a problem with this? What about it?
If you go, like, once you go once around, don't you have to come back past that A second time?
You'd actually, it will either go stop here, or it will somehow bypass this one and go all the way to A sub L. So it won't have to read all the way around again necessarily. Think of it this way. Transcription really is starting from one spot. There are 3 different promoters, but they're all right up here. And we've got 2 poly A tails. What does that mean? We're going to produce potentially how many pre-mRNAs? 2. How many genes do we have? We've got 8 of them. One key feature we've talked about before, eukaryotic ribosomes do not read polycistronic mRNAs. And so we have to think about how does this virus overcome that particular hurdle. Note also, some of these open reading frames are overlapping. For example, E1 overlaps with E2, E2 overlaps with E1, E4, and E5. Okay, so you could think about how there might be some different strategies to deal with these features. Sabrina.
What is AL and AE again?
Those are the poly A signals for the early transcript and for the late transcript. So we'll come back to this, but I just wanted to point out that feature. Here are the 8 proteins. The ones that are highlighted in blue there, so E1, E2, E6, and E7 will be the major ones that we talk about. E4 helps in exit. This is the one that actually helps to overcome actin remodeling. E5 disrupts membrane proteins and actually is involved in inhibiting the immune response by preventing the expression of MHC1 on the surface. And then L1 and L2 we've already talked about are our capsid proteins. E1, helicase involved in replication. E2, numerous roles. We'll talk about each of them. E6 and E7 involved in regulating the cell cycle. These are the major ones that we consider when we talk about cervical cancer. All right, so here's the life cycle. Go ahead, Jonah.
Quick question.
Yeah.
When it says that it disrupts MHC1, is it making it not expressed, or like?
It destabilizes it. So the protein gets made, but since the role of E5 is to disrupt membrane proteins, MHC1 is a membrane protein. So it's expressed, or the mRNA is translated into protein, but that protein loses its three-dimensional conformation, and so never actually makes it to the surface of the cell to present these internal epitopes to T cytotoxic cells.
I feel like I remember from micro, there was a cell that looks to see if cells are lacking.
NK cells.
So if an NK cell finds one of these HPV infected cells,
Quite possible it would kill it. That's a really good point. Now, this area, the area where HPV infects, may be a bit. It may lack some immune response because it's not getting into it. If we take a look at where the infection is occurring, it doesn't pass the basal membrane right here. It's possible that you might get recruitment of NK cells, but I feel like this is a little bit immunoprivileged in that there is an immune response here. That's a really good question, Jonah. Bear with me, this is where we really need Brenda, but so we have here an image showing, this would represent the cell undergoing replication. Here's metaphase. For an infection to occur with HPV, there needs to be a cut or an abrasion, most simply put. And so this route is known as the parenteral route. I'll spell it out real quick, but basically what this means is it's entering through some sort of cut, scrape, etc. And in normal epithelial cells, what we have is a basal layer of cells. This is where we would find our epithelial stem cells located, ones that are able to replenish not only themselves, but to produce the other cells of the epithelium. And as these cells move upward, as they get pushed upward from replication of cells further down, we find differentiation, increased production of keratin, so that these that are at the surface are protective. Mostly at this point, they are not replicating. They're actually dead cells, and those dead cells are the ones that slough off. In the case of HPV, though, it prevents this death from occurring. HPV, on the other hand, is going to encourage replication of these basal epithelial cells, the ones that are bound to the basal membrane, and encourage continual proliferation. And in those keratinized cells, then, what we find is this is where the late proteins get expressed. And ultimately, as those cells are sloughing off, we're also getting release of the viral particles from, in this case, the wart. One of the things to point out here is note that gene expression correlates with the differentiation of these cells. In the most undifferentiated, we find E1, E2, E6, and E7 expressed. As those cells undergo differentiation, we begin to find really a lot of viral replication occurring and expression of E4 and E5, L1, L2. Virus assembles on the most differentiated cells and then releases from there. One of the important things to keep in mind is the virus needs to keep that cell alive. If the cell dies, the virus can't replicate, it can't translate, it can't transcribe the RNA. It's actually dependent on the host for all of those features. Sabrina.
So it must start in the basal layer of the epithelial cells, so if the cut's not deep enough, it can't start in the next layer?
Exactly. Exactly.
Then how do people, cause can't you get like warts on your skin just through contact, like even without a cut?
So I think that you would need, I don't think that's true.
Okay.
You do need a cut, but it doesn't have to necessarily be a visible cut. It can be a microabrasion.
Okay.
So like if we're talking about, this is true for HIV too, and I'm thinking about sexual contact in particular. Like oftentimes after sex there may not be a visible cut, but microabrasions may occur which create a portal of entry for the organism, or for the virus. Jonah.
How does like the outermost layer stay living when it's like that far away from blood vessels or like,
I think part of it is the virus prevents the cellular death from occurring. I don't have a great answer for that. And in part, I may not have a great answer for it because we may not know the answer right now. Like I said, one of the things that's been very challenging with HPV is we haven't had a really good model outside of a living organism to study this. I think we can now start to address those questions, and I just don't know what the answer is.
I just don't know how it would stay alive. Like
That's a great point.
water or
No, that's a really good point. It may be that it's only alive until this point, and then at this point, self-assembly is completely without energy. So the cells may not need to be alive at that point of viral assembly. That might be part of it. Sabrina and then Grace.
How does the virus exit if it doesn't kill us?
Actually, I think it's from my understanding is it's actually from the cell rupturing. So it may not kill the cell early on, but think of the view of a wart. One of the features of a wart is it's kind of rough, which is the keratinized cells, but also those cells are going to break open as well. And that, I think, contributes to the release. Grace?
Okay, so I'm just clarifying. So when differentiation, sorry that was weird, occurs, the late genes start being expressed?
Mm-hmm.
Okay.
And there's a little bit more than just that. There's a couple of things that contribute to this change in gene expression. One is E2 actually is a transcription factor that's going to recruit other proteins. So that's one feature. Another feature is the genes that are normally expressed in a cell in the undifferentiated versus differentiated cells begins to change as well. That can help to change gene expression. And then finally what we find is E2 will perform one more trick. And we'll talk about that last trick in a few minutes. So here are the first three steps: attachment, uncoding, or penetration and uncoding. We know that HPV requires heparin sulfate, which is found in the basal membrane, for attachment. There's thought to be a second receptor. Nobody knows what it is, if there is one that exists. Okay, and so this is still an area of active investigation. We really don't know a lot about these early steps of infection. We do know that the virus, because it is naked, it must enter by endocytosis of some form. That's why we think that there's a second receptor here. The drop in pH will lead to the uncoding of L1, or L1 will detach from the genome in L2. And then L2 interacts with a protein known as SMX17, which is also known as, the full name is, write this down. So the full name of this protein is sorting Nexin 17. It should have no meaning whatsoever to you. but the sorting indicates that it has some sort of role in identifying where that endosome is releasing material. Basically, L2 interacts with this and allows it to escape the endolysosome so that the pH doesn't destroy the DNA. L2 then, at this point, after that uncoating step, will move on microtubules to transport the DNA to the nucleus. Once it's into the nucleus, the role of L2 is done, and now all you've got is the viral genome inside the host cell. Because there are no other proteins, this means the virus has to be reliant on what for transcription to occur? It has to be reliant on the host. HPV does not carry its own RNA polymerase. So I want to come back to our image of the two transcripts that get formed real quick. So in blue here, I'm showing you the early, which would include from the promoter region, would be able to transcribe E6 or E7 or E1 or E2 or E4 or E5. And then the late ones go all the way around, transcribe the full, almost full 8 kb of the genome and can produce L1 and L2. What mechanisms can contribute to this? So what I want to do is, I'm going to give you guys 3 or 4 minutes, and what I want you to do is brainstorm with somebody else about what mechanisms could lead to the production of monocistronic mRNAs here, or would allow for these genes to all be expressed, but simply from 2 potential pre-mRNA transcripts. All right, so think about mechanisms that we've talked about. I'll give you a couple minutes.
You know what I'm talking about? Yeah. What are those? What?
Next week I'm working.
Shanae, I heard you say one possibility.
I said leaky scanning, just because then I could like skip some codec sequences and like go to the next one.
I actually think that would work here, right? This is an area where leaky scanning would actually be a really good possibility. Others?
Frame shift?
Not a bad idea. That also could account for some of this as well. Polyprotein?
Mm-hmm.
Polyprotein could be possible, but none of these account for all of these issues, right? So polyprotein wouldn't necessarily allow for you to produce E7, E1, and E2 at the same exact time. Alternative splicing is the mechanism that actually gets used here. But any of those mechanisms are possibilities, right? And so what we would start off with, or the way to identify this, is we'd take this genome and, for example, look for signs of leaky scanning, or look for signs of, do northern blots to figure out how many mRNA transcripts are actually produced, for example. And this does a lot of alternative splicing. We're not going to cover all the transcripts. I took a look at one paper and I nearly threw up because it gave me quite a bit of anxiety. But I found this and I figured this is a good explanation. So remember what alternative splicing is basically that sometimes an mRNA sequence can be an intron, sometimes it's not an intron. That's basically what we mean by alternative splicing. So here's an example in which we have an intron located here, or a potential intron located here, intron here, and another region that sometimes is an intron, sometimes is an exon, depending on the expression of different factors. Those factors can be what are known as heterogeneous nuclear ribonuclear proteins, or HNRNPs. They repress alternative splicing. The others are what are known as SR proteins, or serine-arginine proteins. They're rich in those two amino acids. E2 actually will bind to serine-arginine proteins and direct where splicing is going to occur. Different serine-arginine proteins will recognize different splice sites and will then recruit the different small nuclear ribonuclear proteins, or SNRNPs, U1 and U2, to different regions for splicing to occur. I'm not concerned with you knowing the mechanism, okay? What I do want you to know is alternative splicing explains the way in which we have two potential pre-mRNAs that can produce a variety of different mature mRNAs. Sabrina.
So the first mRNA made contains those multiple, like E1, E2, all that stuff in there, and then the splicing splits it into the individual mRNA.
The splicing, what it would do is it would leave each of those transcripts with most likely only one open reading frame that can be transcribed. And so the one that produces, for example, the E1 would look, that mature mRNA would look different than the mature mRNA that produces E7. I think I may, yeah, here's the picture of this nonsense. And this isn't even the worst of it, but this is just showing from two different promoter regions transcripts that will produce E1. And look at how much for E1. It also keeps the sequence for E2, E4, and E5 around as well, but only this open reading frame for E1. So from here to here contains the open reading frame that would actually be translated. Right? The rest of that sequence would just be part of the 5' untranslated region or the 3' untranslated region. For E6, note that this would contain open reading frame for E6 here, but also contains the coding sequence technically for E7 and also for E4 and E5. But E4 and E5 are not translated. They are now part of that 3' untranslated region. Okay. So there is a different one of these for every single one. I'm not really sure what the E1, E4 one is, so don't ask me about it. But the rest of these, all created by alternative splicing. And again, the protein that contributes to this then is E2. E2 is regulating which of these different spliced mRNAs is going to be created. And it does it by recruiting or interacting with the different serine-arginine proteins, or those SR proteins. We do find some role for repression of splicing as well. So in undifferentiated epithelial cells, the creation of the L1 mRNA is repressed, or the creation of or splicing of the L1 mRNA is repressed by those HNRNPs. And as their expression drops during differentiation of those epithelial cells, now we find L1 being expressed. All right. Okay. One thing I want to show you, and then one last thing to do for today. Let me see if I can find this real quick. No, that's not. So I mentioned one of the challenges right now, or one of the challenges up until maybe a couple of years or a couple of decades ago. Alright, here we go. So one of the challenges to a couple of years ago, or decades, sorry, was how do we actually grow HPV in cell culture and watch this process through differentiation of epithelial cells? The method that's used now is known as a raft culture, R-A-F-T. And in this, what you do is you first take your cells, you grow them at the bottom of a plate. Once they've reached confluency, and these would be epithelial cells or human keratinocytes, you move them to a metal grate, which now serves as your basal membrane. And you put that metal grate or supportive grid with culture media just below it. And now, applying the right growth factors, you will find these cells begin to differentiate as they move upward in this raft culture. And here is what those raft cultures look like. And you actually can see here, here's the basal layer. And as you move upward, you're starting to get the keratinocytes produced here at the top. So this does a pretty good job of reproducing what we find in vivo. And using this technique, then, we can start to manipulate HPV in cell culture and, for example, study things like, okay, so how does this escape actually work in these keratinocytes? Took a long time for people to develop this method, but the use of it over mouse culture to study HPV has been kind of a game changer for studying HPV. Amelia.
What's the reasoning for the metal underneath it?
I think that serves kind of like the same purpose as the basal membrane. It's just there to, so you can imagine it as being like, the metal's going to be coated with heparin sulfate and other connective molecules that would allow for those epithelial cells to attach to it. And just having it on this surface here then allows you to kind of have those cells hovering above where the growth media is at. Yeah. So you could kind of think of it as like just below where that is where your bloodstream would be located, or a blood vessel that's carrying nutrients for those cells. All right, last thing for today. Well, first, any questions before I go any further? Okay, grab a sheet of paper.
Oh.
What?
Can we split with a buddy?
Sure, you can split with a buddy or an enemy. It's fine. Paige, you don't have to do this. If you don't want to rip out of your notebook, you don't have to do this. Well, you can. I'm just saying if you don't want to rip out of your notebook, oh my God. Okay. All right, here's what I want you to answer. So the big point today was talking about how does HPV overcome a host cell hurdle. In particular, how does it overcome that hurdle of
Do we have to put our name on it?
Nope.
Okay, good.
How does it overcome this hurdle here? So compatibility of the viral mRNA with the ribosome. We said it has 2 pre-mRNAs, but it encodes 8 genes. What I want you to do is just write down, how does it do that? Okay, to the best of your ability. Do you need me to repeat, Sabrina? How does it overcome the host cell hurdle that those mRNAs need to be monocistronic? And then once you are done, feel free to pack up and head out, and I'll see you all on Wednesday.